Good morning. Today we are going to review the cell cycle, in particular to mitosis, and then we're going to complete an activity where we create a foldable visual guide that demonstrates our understanding of the process. Let's go. Okay, we are going to do a quick review of cell division before we go into the activity. Can someone recall from our last lesson what the goal of cell division is? Yes, Tucker. Okay. It's where a cell makes a copy of itself. Great. Let's see. Remember that cell division is the process by which cellular material is divided between two new daughter cells. So the mother cell becomes two daughter cells. The two daughter cells will be identical to each other and to the mother cell. This happens when each daughter is half the size of the parent cell but immediately begins growing. So a typical human cell has about 2 meters of DNA before the cell can divide. All of this DNA must be copied and then the two copies separated so that each daughter cell ends up with a complete set of DNA. Each species has a characteristic number of chromosomes in each cell. Chromosomes during eukaryotic cell division. Each cell must first copy its chromosomes before cell division occurs. Each daughter cell gets a complete copy of that information. Cell division occurs in two main stages: mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. Remember, interphase includes the G1 phase, the S phase, and the G2 phase. It is the period of time preceding mitosis. And mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. Again, this is an overview. We've been covering this process in the last couple lessons, so we just wanted to review it before we went into our activity. Now, let's move on to the instructions for our cell cycle foldable guide. We are going to start with the folding and cutting. Everyone should have a piece of paper in front of them. First, what you're first going to do, you should have a piece of paper that looks like this. You're going to fold this into a trifold. You put each side together until it touches and crease the edge. So, it should look like this, a 3-part fold. Okay, after that, you measure the paper and cut the fold so that there are 4 flaps on each side. Your foldable should look like the picture to the right. So, on each side, you measure it and you cut with your scissors 3 different cuts right there, and you're going to have 4 flaps on each side. Then we will move on to labeling. So on the top tab on the right side, you're going to label the G1 phase, like a title. You underline that. And then right underneath that in the tab, you label it the S phase. Underneath that in the tab, you're going to label the G2 phase, and then at the bottom, you'll have the prophase. Then next, you're going to move on to the left-hand side. On the very bottom, you label it the metaphase. Then on top of that, you're going to label the anaphase. Then the telophase above that, and finally at the very top on the left-hand side, cytokinesis. Then on the inside, that's when you take the paper and you fold it. You unfold it. Ah, there. That's better. So it should look like this, 3 parts that are folded together. On the very, very middle, you're making, just in the middle section, you're making this drawing of the cell cycle. So it's going to look like this. You make a circle. You're going to put the S1 phase with an arrow to the S phase, the G2 phase, And then the final phase will have the sections of mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis. Then we'll move on to the drawing. You're going to draw a picture of what the cell looks like in each phase of the cell cycle on the outside of the tabs, under the titles you wrote earlier. So, the G1 phase, you'll have a little picture of that phase right here. Then there's a picture of the S phase, the G2 phase, and the prophase. Notice you can see the chromosomes separating, getting ready to flip over. And then in the metaphase, they line up. You can see the spindles. And then they'll separate in the anaphase. Make sure these are as detailed as you can make them. And then the telophase, you'll see the two cells starting to merge away from each other. And finally, in cytokinesis, you have two identical cells. Remember, on the inside of the tabs, on the right side, you're going to write a brief description of what happens during those phases. Then you'll also do that on the left-hand side. I'll show you a picture of that in a minute. Then we'll move on to coloring. So, you're going to color the G1, the S, and the G2 sections in light blue. You can see it right here. And then you're going to color the phases of mitosis in orange. It might be hard to see this, but these are shaded over with an orange, just an orange colored pencil. And then you're going to use a colored pencil on these three phases in blue. This is what it's going to look like. So this side of the outside is going to be colored blue as well. And then this side of the outside tabs are going to be in orange. So you'll have blue tabs on the right-hand side and orange tabs on the left-hand side. When you open it up, you'll have a circle of the cell cycle in the middle. The G1, S, and G2 are in light blue, and the stages of mitosis are colored in orange. Then there's a description of each phase. Remember the top on the G1 phase? On the inside of that tab, you're going to write cell grows and prepares for division. On the inside of the S phase, you write something like DNA replicates or makes a copy of itself. Then you're going to do that with every other phase on the inside of the tab. And then finally, we'll get to coloring the outside. You're going to color all of the chromosomes in purple. Those are on the inside of the nucleus. You could just kind of outline them so that they pop out. There's not much to color. You're just going to shade them in. Then the cytoplasm will be in yellow. That's the inside. The cell membrane will be colored in red. Again, that's just a line, so you're going to make it really, really dark, a dark red line. And then finally, the nuclear membrane will be colored in green. Okay, you can go ahead and work in your lab group on this. There are colored pencils on the tables, and then I will walk around to offer assistance. Thanks.